You know, there's a hidden crisis unfolding right under our feet, in the very soil that grows our food. It's a silent threat for sure. And this is the story of the incredible race against time to save our vegetables.
33%. Just let that number sink in. That's how much of our planet's most productive, irrigated farmland is being threatened by salt. And look, this isn't some tiny problem on the horizon. It is a direct threat to our global food supply, and it's happening right now.
And the culprit? It's something as simple as salt. This isn't a problem for the future. It's silently poisoning our best farmland as we speak. But scientists are fighting back, and their race for a solution is this fascinating story of genetics, ingenuity, and a little bit of hope.
Okay, so before we get into the really cool solutions, let's first get a handle on the sheer scale of this global salty crisis. It's big.
So, what exactly are we talking about when we say soil salinity? Well, in simple terms, it's when salts, like sodium and chloride, build up in the soil to toxic levels. This essentially poisons the ground, making it nearly impossible for most plants to drink up the water and nutrients they desperately need to survive. And here's the scary part. This problem is growing. We're losing about 1.5 million hectares of good farmland every single year. A lot of this is because of poor irrigation practices, where water evaporates but leaves behind this devastating concentration of salt. And trust me, this isn't just some abstract number on a global report. We can see it on the ground. You look at places like India, and huge agricultural states like Gujarat are getting hit incredibly hard. This is the front line of the crisis. All right, so we've seen the big picture. Now let's zoom way in, right down to a single plant. Because salt doesn't just make it a little harder for a plant to grow, no, it attacks it with a devastating 1-2 punch. So the first hit is something called osmotic stress. The high salt in the soil basically makes it incredibly difficult for the plant's roots to pull up water. It's this cruel irony, right? The plant is literally dying of thirst while it's sitting in wet soil. Then comes the second punch, ionic toxicity. The salt ions that do manage to sneak in start to build up, slowly poisoning the plant's cells and shutting down its most critical functions.
And what you end up with? Well, it's a world of difference. Instead of a healthy, thriving plant, you get one that's stunted. Its leaves are burned, and the fruit it produces is just sad. For a farmer, it's a total disaster. And for the rest of us, it means less food on the table.
But here's where things get interesting. Nature is clever. Over thousands and thousands of years, some plants have actually evolved their own defense playbook to survive in these super salty environments. And understanding this, well, it's the key to everything.
This quote right here really gets to the heart of it. You see, true salt tolerance isn't just about building a better wall to keep the salt out. It's all about having a smart internal system to manage the salt that inevitably gets inside.
So plants basically have three main strategies. First up is exclusion, where they actively pump salt ions back out of their roots. Next, there's retrieval. They literally pull salt out of the xylem, that's the plant's plumbing system, before it can reach the sensitive leaves. And the final trick? Sequestration. This is where they lock up those toxic ions in a kind of cellular storage bin called the vacuole, keeping them safely away from the rest of the cell. Pretty smart, huh?
And that brings us to the core of our story today, the incredible race by scientists to take these natural defense mechanisms and breed them into our most important vegetable crops.
And the tools for this race have evolved, just dramatically. If you go back to the 80s, it was all conventional breeding. Kind of slow, not super precise. Then in the 2000s, we got way smarter, using DNA markers to guide us. And today? Well, today we have revolutionary tools like genome editing that have completely changed the game.
Yeah, the old methods, it was like trying to perform surgery with a sledgehammer. They were just too slow, too imprecise. It could take more than a decade to develop a single new variety. And often, when you'd bring in a useful gene from a wild relative, you'd also drag in a bunch of unwanted traits, like making the fruit small or bitter. They call that linkage drag. Marker-assisted selection was an absolute game changer. All of a sudden, breeders could test a plant's DNA when it was just a tiny seedling, instead of waiting years for it to grow up. It basically turned a huge guessing game into a real science and saved so much time.
And now we have CRISPR. Seriously, think of it like the find and replace function in a Word document, but for DNA. It lets scientists make these incredibly precise changes to a plant's genome, turning on a helpful gene here, turning off a bad one there.
And all without any of that problematic linkage drag. It's amazing.
So how does all this come together? Well, the future isn't about using just one of these tools by itself. It's about combining them into this super powerful accelerated toolkit for modern breeders.
A key part of this new toolkit is something called speed breeding. By basically manipulating light, giving plants up to 22 hours of it a day, scientists can trick them into growing way faster. This lets them cycle through 4, 5, even 6 generations in a single year, instead of the usual 1 or 2 you'd get in a field.
And the impact is just staggering. A process that used to take a year and a half, like creating a stable, genetically edited plant, that can now be done in just 6 months. This is the fast-forward button breeders have been dreaming of to keep pace with our changing climate.
But hey, changing the genes is only half the battle, right? You have to be able to measure the results just as fast. And that's where high-throughput phenotyping, or HTP, comes in. Think of it as giving a super-fast, high-tech checkup to thousands of plants at once, using these advanced cameras to measure stress in ways the human eye could never see.
And this technology is leading to some stunning breakthroughs. For example, a recent study on tomatoes revealed something that completely flipped all the assumptions on their head. It turns out the most salt-tolerant plants, they weren't the ones that built the best walls to keep salt out. No, they were the ones that just kept on growing, efficiently managing their water in spite of the salt. This is a massive paradigm shift, and it completely changes what breeders are even looking for. Which, of course, brings us to the final critical question. The tools we have today are more powerful than anything we've ever had before, combining genetic precision with just incredible speed. But the challenges, from soil salinization to climate change, they're accelerating too. The race is on, and really, the future of our food is hanging in the balance.